Okay, all right. So, good afternoon. We are going to be completing the 2021 paper discussion today. Okay, so I think we have done some of the MCQs. Okay, particularly, I think we finished animal form MCQs. Yeah, in the MCQ paper, and some of the other things, other MCQs are remaining. I think we finished plant form and evolution also, ne, out of the MCQs. Yeah. So today I thought we'll get a start with our second MCQ onwards because in 2021 it was half of the, I mean, one third of the second structure was from plant form, and then from I think part B onwards it was animal form. We'll so start with the structure as a discussion so that yeah, as the beginning. Then we'll later move on to the other molecular and bit of other topics in the MCQ as well, and then we'll complete that. Then if we have time, we'll move on to 2022 paper. Okay. So as you all are here, right, online people, you can hear me now, right? Yes. So we have about 3 more weeks to including today, so 2 more weeks of our usual revision class time. So in that revision class time also, I'm going to be doing now as our final tute. We have completed applied, and it's going to be all past paper based revision from this point onwards. I told you the importance of adapting to the past paper structure at this point, because that's the paper you are going to go and meet in the exam. Yeah. So you have to get more and more familiar with that structure, and then in the exam you will not have that surprised element. You know the paper in and out. You know how it's going to come. You just need to touch up on your knowledge now, right? So that you can go and sort of face it. Okay. So you don't have that terror. You won't be surprised. Okay, this many animal form questions are there. This much plant form, right? That's because in these final days you train your mind to the format in the past papers. I suggest you do to the other two subjects as well, particularly for chemistry. It's mainly patterns of MCQs coming again and again. So if you have a sort of a practice of the patterns that are coming, you can save a lot of time as well as sort of the stress element goes away in the exam because you are not surprised. Okay, is this the question? Is this the way the questions are going to come? You won't be surprised like that. Okay, so today is going to be 21 paper. Then this Friday in the paper class also, it's going to be past papers 22, 23, 24. We'll try to finish 25 as well. And at the end of this month, our sort of the entire class program will be over. Okay. Then in the sort of the next month, first week, I will try to have some guest essays, but I will try to incorporate some guest essays in our Friday paper discussion as well. So some of the guest essay topics that I'm thinking that might come, but that shouldn't deviate you from your normal study plan. Guest essays විතරක් එනවා කියලා ඉවරලා don't think about that. It is again a way to sort of push you to study a bit more, right? Okay, this could be a likely topic කියලා. I'm going to sort of persuade you to study more and more, right? මේකත් වෙන්න පුළුවන්. Everything can come. Yeah. So it's the main purpose of giving you guest essays is not to sort of so that it exactly comes like that in the paper. But again, whenever you are doing a guest essay, you are again studying. So there is going to be another motivation for you to study. Okay, this might come. This is a guest essay. So you are studying that. So that program is also going to happen this Friday. So today we'll finish the 2021 paper so that we are one step closer to finishing these past papers. Okay, right. Shall we come to the second SCQ onwards? Yeah. Second SCQ onwards, online people. Okay. Right. So we'll start there. No? I'll discuss some of the theory parts around the questions as we go along. Okay. So let's see. What is the property of water that helps in transporting dissolved minerals through vascular tissues in plants? Yeah. So they are asking simply a property of water, right? By this word, you should understand how many, I mean, you should understand they are referencing to how many properties. I mean, so under the word properties of water for life, you only learned 4 things. There's cohesive behavior, ability to moderate temperature, versatility as a solvent, and what is the other one? Expansion upon freezing. Yeah, those four. So it has to be something out of those four that you have to write, because they are asking the property of water, right? So what is the property of water helping the upward movement in vascular tissues? Yeah, it is a cohesive behavior, right? That is the due to the hydrogen bonds. Yeah, the cohesive behavior due to the hydrogen bonds. Yes, if you have written according to the, so cohesive behavior is the best one, but they have given in the marking scheme for saying H bonds. Yes, hydrogen bonds, they have given the attraction between motor molecules due to H bonds. But I recommend whenever you see this property, you try, the best, safest choice is to write one of those four. Yeah, those four properties of water vital for life. Yeah, so better to stick to those properties. Okay, so name a protein that has defensive roles. Okay, so the second question also we have some let's say even second lesson things are there, no properties, biological molecules, protein that has a defensive role. What do you call the family of proteins that has a defensive role? You can call them antibodies or immunoglobulins. Yeah, defensive proteins. Who secretes immunoglobulins? Who secretes immunoglobulins? What type of cell? Yeah, what type of cell secretes immunoglobulins? Yeah, in your immunity, in your acquired immunity, there are two types of immunity. In your acquired immunity, you have two types of cells, neither as T cells and B cells. Neither B cells or B lymphocytes, we always say there are two main forms in a sort of a this sort of what we say as acquired immune response. There are two things that they can do. One thing is they respond right now to the problem that they have by differentiating into what we call as effector cells. By differentiating into effector cells. The other thing is they have a memory of what happened, who attacked you. So we call that immunological memory. We call that immunological memory. So those two aspects are there. Got it? So what are the two cells involved in those two aspects? I'll draw an arrow here. What is the effector cell? Who is the working cell or the defensive cell form of your B cells? That is going to secrete this guy. They're called plasma cells. Right? Plasma cells. Okay. Plasma cells. Then who are the cells maintaining the memory? Who is going to remember who attacked me? Yeah. Simple enough. Right? So they are B cells, no? They are simply memory B cells. Simply memory B cells. Yeah? Okay. So who secretes antibodies then? Plasma cells. Yeah. So even in your own paper, you can write a small, let's say, small theory points that is going to help you to remember the theory around this. Okay. So Tagala, just about T cells. What is the? I'm not going to write. What is the effector cell form of the T cell? Cytotoxic T cell. Then of course they have memory T cells as well. Okay. Right. Monomer of the polysaccharide component in fungal cell wall. So remember, they are asking Tagala. You are not going. Some people might directly write what is the polysaccharide. Are they asking that? Okay. First of all, tell me what is the polysaccharide found in fungal cell wall? Yeah, fungal cell wall and arthropod exoskeleton, basic things. Chitin, no? Chitin. Then they are asking not chitin. They are asking what? They are asking the monomer of chitin. Can you see? So what is the monomer of chitin? Glucosamine. These are direct theory points. There's nothing to explain. You have to remember, I think, chitin, it's glucosamine. Okay. Okay, next question. State an event that occurs in mitosis and meiosis 2 but does not occur in meiosis 1. Occurs in mitosis and meiosis 2. In the cell cycle, okay, so mitosis is self-explanatory, right? What happens in mitosis? Yeah, you have a cell that is just going to divide into two cells, no? Yeah, so the first cell is called the mother cell, is also diploid. and the daughters are also going to be diploid. It's just a photocopying process. Ne? Yeah. So before this mother cell divides into these, it's going to have how many copies? It's going to have 4 copies. Ne? Because by the DNA copying duplication process, it's going to, this 2n is going to be doubled during the which phase? S phase. Ne? So then in mitosis, what's going to happen? This newly copied DNA will be sent to the daughter cells. Then in meiosis, I always like to say that, so meiosis has 2 parts. No? Meiosis has 2 parts. What are those parts? They have meiosis 1 and meiosis 2. Meiosis 1, we call it as a reduction division. Why do you call it as reduction division? Meiosis 1, why do we call it as reduction division? That is because in the mother cell in meiosis, this is the place where the mother cell becomes haploid. Okay, because of the separation of the homologous. Our mother and father's pairs are separated in meiosis 1, no? Right? Meiosis 2, we describe it as similar to mitosis. Right? Meiosis 2 is similar to mitosis. Why is that? Because that is where the sisters separate. So, you know, if you take a cell that is about to divide by meiosis, this is the sister chromatids. No? These are the 2 sister chromatids that are connected at their centromeres. In meiosis, something interesting happens. No? The same similar pair from your father. So the similar pair from your father. Why do we call that? The homologous chromosome. They will pair up. You know this. What do you call this connection between these mothers and fathers chromosomes? That is called what? Their connection is called synapsis. You know, the synapsis. So this synapsis happens during meiosis 1. Then this blue and reds will separate in meiosis 1, and in meiosis 2, what's going to happen? These sisters are going to separate. The blue and blue are going to separate. So, as I just mentioned, what is the similarity between mitosis and meiosis 2? Separation of sister chromatids. Yes, separation of sister chromatids. Because in meiosis 1, the sisters stay together. You can say just chromatids or separation of sister chromatids. Yes. Okay. State where CO2 is first fixed in C4 plants. CO2 fixation in C4. Yeah. C4 plants have a separate carbon dioxide concentrating mechanism. CO2 concentrating mechanism is there. I can relate it that to the next question as well. PEP carboxylase in the C4 of photosynthesis is more efficient than RUBP carboxylase in C3. Yes. What can you say about this? Okay, I'll give you a quick reminder of the C4 pathway, right? Final time, huh? Final time. Okay, final time for the C4 pathway. Okay, what's going to happen? There are 2 types of cells. 2 types of cells are involved in the C4 pathway. You have your photosynthetic cell. What is the photosynthetic cell? The cell in C4 plants that are nicely designed to do photosynthesis, the light reaction. The light reaction, they are the mesophyll cells. So I'll draw them in this green color. Then you have the cell that is designed to do the Calvin cycle. Right? That is called the bundle sheath cell. So those 2 cells are there. So the photosynthesis is divided into 2. The process is distributed between 2 separate cells. So you have CO2 gas that is being absorbed into the stomata, and then it enters into the mesophyll cells. There's a person ready to accept the CO2. That is the first carbon acceptor, that is the phosphoenol pyruvate. Phosphoenol pyruvate will accept this PEP and form what? Oxaloacetate. This reaction is catalyzed by what enzyme? PEP carboxylase. Then this oxaloacetate will convert into some other intermediate products such as malate. Okay, then it will diffuse. I'm just giving a rough, quick sketch. Okay, then it will convert back into oxaloacetate. Then, as it's giving back the CO2, it will convert back into phosphoenol pyruvate, and this PEP will diffuse back into the mesophyll cells. So, as the same at the same time, this CO2 is released. The CO2 will enter into the Calvin cycle. Who is the enzyme waiting here to accept this CO2? There is Rubisco. What is the long name of Rubisco? RUBP, carboxylase oxygenase. So that then it will produce our normal Calvin cycle products. What is the first carbon containing one? 3-PGA. So that is the Calvin cycle. Okay. So what are they asking? CO2 is first fixed in plants. That is this cell, no? Yeah. This green or the where the light reaction is happening is the mesophyll cells. Yeah. Light reaction is happening in the mesophyll cells. Okay. Then two reasons for PEP carboxylase being more efficient than RUBP carboxylase. Okay. Why is this guy more efficient in C4? That is because I didn't mention this step. This carbon dioxide is not actually being it's not reacting with a PEP as carbon dioxide. There's an intermediate reaction. What is that intermediate reaction? That CO2 is actually converted to bicarbonate before it is reacting with the phosphoenol pyruvate. There's an enzyme here that is called carbonic anhydrase that is also found in where? There's an enzyme here. You don't need to know that, but it's actually called carbonic anhydrase. It does the exact same reaction somewhere else, but not in a plant. This reaction, okay, listen, this reaction of converting CO2 plus H2O, okay, reversible reaction to H2CO3, and that will convert into H plus bicarbonate. No, this is a stepwise process. This reaction is done by an enzyme called carbonic anhydrase. Yeah, it is also found in where? Be more specific. It is also found in your red blood cells. It is also found in your red blood cells. Okay, because in red blood cells, similarly in red blood cells, CO2 will diffuse into this red blood cell, and there's the same enzyme there. Then it will be converted to bicarbonate. Actually, bicarbonate comes out of the red blood cell, and it will just travel in blood in aqueous form. That is one of the ways carbon dioxide is transported. What percentage? What is the percentage that you transport as bicarbonate in blood? Yeah. What are the three ways that you can transport carbon dioxide? Okay, I'm getting very further from the thing we are discussing, but taggala here. What are the ways that you can transport carbon dioxide in your blood? You can transport it as this 70%. Then you can transport it as bound to hemoglobin as carb amino hemoglobin. Okay, carb amino hemoglobin. How much is it? 23%. Then what is the remaining? How much is remaining? 70 plus 23. You have 93% remaining. Sorry, 7% remaining. The rest is it's just going to be CO2. I mean aqueous. That means it's a free gas. Okay, because CO2 is slightly dissoluble, right? As free gas, 7%. Okay, right? This is the C4 pathway. Got it? The C4 pathway, as I was saying, the phosphoenol pyruvate is more efficient than this guy. This PEP is more efficient than Rubisco. One thing, it is not reacting with CO2, but it is reacting with bicarbonate. So PEP carboxylase actually has a higher affinity to bicarbonate than CO2. Okay. Then the other thing is, unlike Rubisco, so Rubisco ke full name hai what? RUBP, carboxylase as well as oxygenase, because he likes oxygen as well. Okay, because whenever if he has oxygen, he will react with oxygen. If you have oxygen here, he will react with oxygen as well. That is the photorespiration process. But pep carboxylase is not like that. Pep carboxylase doesn't like oxygen. He doesn't like oxygen, so we don't have to worry about any oxygenase reaction happening. So you can say in that question, okay, what can you say? You can give so mesophyll cell first one. Then you can say what are the reasons pep carboxylase is more efficient. One, it reacts with bicarbonate, not CO2. It reacts with bicarbonate. So similarly, you can say it has a higher affinity. Pep carboxylase has a higher affinity to bicarbonate as well. Then the other thing is, the most important one actually, pep carboxylase has no affinity, or you can say does not react. No affinity for oxygen. Has no affinity for oxygen, or doesn't react with oxygen. So those two reasons make it better. I mean, so pep carboxylase is a very good carboxylating enzyme compared to Rubisco. Okay, that's why overall the C4 pathway is much more efficient. That's why the C4 pathway is overall much more efficient, because it's the upgraded version. Yeah. Okay. We'll quickly finish this. Then there's a direct definition question. That's why I think I have been doing this for my 27, 28 people. I have advised them to highlight all the definitions in just one fixed color. Yeah, all the definitions in one fixed color. So whenever you turn a page in the resource book, you see the definition in a highlighted way. So if you have done that, then it's good. Okay, because they are asking now the direct textbook definition here. Okay, so in secondary growth, you can see they have given 3 rules to write. Okay, so you have to say it is an increase in diameter, of course. Increase in diameter, no? It's how it's how the plant get fat, right? Increase in diameter. It happens in stems and roots. Okay, increase in diameter in stems and roots due to new cells, no? Produced by who? Produced by, you have to mention produced by who? Vascular cambium, right? Vascular cambium and what else? Yeah, and cork cambium. Or you can just say what is the other term that you can give for these two cambia. Yeah, if you take the plant, they increase your lateral diameter. Plant, if the plant's primary structure was like this, at the end you will have a lateral diameter increase, no? Lateral. So you can call them as lateral cambia, right? So you can either list the both two out like this, which is something good, okay? Or you can just say produce new cells produced due to lateral meristems, okay? Due to lateral meristems, okay? That's a textbook definition. So you have to mention it is increase in diameter. They have given 2 marks for this writing this answer. So it is going to be increase in diameter due to either these two cambiums you have to write, or you have to mention it's a lateral meristem. Okay. What are the other two types of meristems? You have apical meristem that are making the plant long. Right? Apical meristems are of two types as shoot apex, root apex. Then if you take like something like bamboo, never grass species, in resource book it's given certain grass species. What do you have in between your nodes? What do you have? This is a type of meristem you find between nodes. Yeah. And in between nodes, from there you can get some leaves as well. Yeah, what do you call those? Intercalary, no? Intercalary meristem. Intercalary meristem. There is also another type of meristem. So you have lateral meristems, apical meristems, and intercalary meristems based on location, actually. Okay, based on location. Okay, we'll move on to a bit of stomatal opening. Two factors that are responsible for opening of stomata other than light. Okay, so have they eliminated the light factor, right? What else? Yes. What else can open stomata? What else can open stomata? Yes. Stomata is the main venue that you can. Yes, for photosynthesis, it's the main sort of ventilation opening, right? So, in order to perform photosynthesis, what is the main thing you need? A plant needs CO2, no? Because it's one of the reactants. Yes, water and CO2. You have to add water and CO2, right? So, when the CO2 concentration decreases, or you can write decrease in CO2 concentration where? The space just above the stomata, no? What do you call that? CO2 CO2 decrease in substomatal cavity. Yeah, substomatal cavity. Then, of course, I mean, it is kind of related to the light, okay? That is the internal clock, okay? The second one you can write internal clock. Internal clock, where? In the guard cells. In guard cells, okay? Then, something you need to practice drawing, right? So, when you're studying for the final time, something you need to practice drawing is to be able to draw and quickly describe a dicot leaf cross section. You should be able to quickly draw and describe a dicot leaf cross section. The monocot leaf is also important. Okay, that diagram is also there. No? Okay, so you should be able to quickly draw and describe, right? Then what are the things you can see in the dicot leaf cross section? Of course, you can see mainly on the underside. How is the dicot? Mainly on the underside, you can see what? Stomata. Yes. Stomata, when you are drawing stomata, why did I draw the guard cells in green color? They are the only photosynthetic epidermal cells. Ne? So, if I draw another stomata here, right? They are the only photosynthetic epidermal cells. Yes. This is the epidermis. This is the epidermis. Okay. Then usually above the epidermis, what do you have? On the surface of epidermis, you have a small cuticle, layer of cuticle as well. Or waxy. Yeah, waxy cuticle. Got it? You have a bit of waxy cuticle as well. It's found on the underside of the leaf as well. Then if you quickly draw the sort of the arrangement of your arrangement of your leaf parenchyma, immediately under your upper epidermis, what do you have? You have your special type of mesophyll cells that you call as, yeah, what do you call these? The tightly arranged layer of mesophyll cells that is immediately under the epidermis, upper epidermis. What do you call these mesophills? They are called palisade mesophills. They have a very high density of chloroplasts. Their chloroplast density is very high. Okay, palisade mesophills. These are dicot cell. Okay, then you have loosely arranged mesophills. They are called what? Spongy. Yeah, they are sort of not very tightly arranged. Spongy mesophills and palisade mesophills. This is what we call as the substomatal cavity. Substomatal cavity can have evaporized water from these mesophills, right? Water evaporating from these mesophills can be there. Okay, that is one of the main sites of transpiration. You need to remember these theory points. Okay, so Whenever the CO2 concentration, listen, whenever the CO2 concentration here decreases, that acts as a signal for these guard cells to open up the stomata so that CO2 can be taken into the substomatal cavity and can be used for photosynthesis. Okay. Then also there's a guard cell. Then if they asked you, so particularly they are asking two factors responsible for opening of stomata. Can, if they ask controlling stomatal opening and closing කියලා තිබ්බා. Opening and closing both. What is something that you can add here? Yes, yes. There's a signaling, plant growth regulator that we call as ABA. ABA has a direct action on the guard cell, sort of directly ordering him to close, because ABA is secreted when the water concentration is very low. Yeah, water availability is very low. So there is another special thing that is mainly about closing the stomata. Okay, clear? So today itself, have a rough overview of the structure of the dicot leaf together with the monocot leaf as well. I just reminded you. Okay. Okay, let's move on. What is the special feature of soil in which Nepenthes is grown? Nepenthes. Nepenthes is what? Nepenthes, Drosera, and what is the other example that you have? Nepenthes, Drosera. Online people, what is the other plant that you have under? What am I talking about here? Carnivorous. Special nutritional mode of nutrition, carnivorous plants. Yes, Nepenthes, Drosera, utricularia. Nepenthes. I can't remember if Nepenthes is the pitcher plant. Is it the pitcher plant? Nepenthes. Or is it Drosera? Somehow, they are your carnivorous plants. No, they are designed to sort of catch or sort of trick insects. Catch or trick insects so that they are getting caught in their special structures, and those insects will be digested by enzymes to obtain what? Usually nitrogen. So now we have done molecular, sorry, microbiology. You know what is the most limiting nutrient in soil? What are the two most limiting nutrients in soil for a plant? Two most limiting nutrients in the soil for a plant. Phosphorus is the most limiting one, nitrogen, right? That's why we apply so many nitrogen containing fertilizers. Yeah. So many plants face nitrogen deficiency. So particularly if you are living in a soil where there are no microorganisms, okay, these sort of plants live in sort of acidic soils, could be sort of muddy soils, right? So the soil doesn't have a lot of microorganisms to fix the nitrogen for them. Therefore, they have no other option but to obtain it by digesting sort of the bodies of insects. So the body of an insect contains amino acids, proteins. It could contain what? Certain chitinous things are there. No? So they, from that they can obtain nitrogen. Okay. So Nepenthes is a carnivorous plant. Remember. Then what is the problem there? The soil is poor in mainly nitrogen. It could be some other things as well. Could be calcium, could be iron. Okay, other essential minerals could be deficient. So by digesting an insect, you can obtain those, right? Magnesium, calcium, got it? Poor in nitrogen and other minerals. Is it Drosera the Bandura? Have you heard the pitcher plant? No, Drosera is the sundew. Drosera has this sort of a bit of a glue that is like that looks like nectar, right? Drosera. Drosera has this sort of a glue-like thing, yeah, that is going to sort of make the insects get stuck. Then Nepenthes is what? Yeah, Nepenthes is the pitcher plant, right? So the insects is just going to fall down into this, and this sort of the bottom of this pitcher is full of enzymes. Yeah, So the insect is going to be slowly digested. The sort of the mouth of this pitcher is kind of slippery, as well as it has some nectar. So the insect is just going to walk around it, and then it's going to slip into it, and then it will be digested. Anyway. Okay, what happens to the triploid nucleus formed after double fertilization? Okay, I think angiosperm anthophyte life cycle we have gone through so many times. Right? The flowering plant life cycle we have gone through so many times. Something unique about the anthophyte or the flowering plant life cycle is called double fertilization. No, I have talked about this a lot. You get two sperms. You get two sperms. One sperm will of course join with the egg, and that will become the zygote. That is okay. That is one fertilization of the double fertilization. What's going to happen to the other egg? So the other sperm. Okay, what's going to happen to the other sperm? They are going to fuse with the two polar nuclei. Polar nuclei. So two polar nuclei plus one sperm, you are going to get three ends, three copies. Then that is going to become your triploid, no? 3N nucleus. Yes. So what's going to happen to this 3N nucleus? What's going to happen to this triploid nucleus? It is going to develop into what? Yeah. Yes, endosperm. Endosperm is the food source. So if you are having a jack seed, there's like a starchy part there. Piti kossata you boil and eat and what you cook and eat. Jack seeds. The starchy part that you are eating is the food source for the embryo that is coming from the zygote. So that endosperm was the triploid nucleus. Got it? Keep that in mind. Specific location of statoliths. Can you see it's a bit here and there, but there it's going to cover the entire plant form and function lesson. Okay. Specific location of statoliths. Statoliths. So the liths usually refer to sort of stones. Okay. So stato means starchy stones. Okay. Statoliths. Lithasis, cholecysthasis, if you have stones in your gallbladder. Okay. So lith is a stone. Okay. So statolith is a starchy stone. So a statolith is composed of starch and it's a dense granule. What do you mean by density? That means it feels gravity, something is dense. Okay. So what is the specific location? They are asking the very specific location. You have to say in root cap. In the root cap. In the root cap. So that is how, what is it involved? So you don't need to write this. What is it involved, these statoliths? Can anyone tell me? What is it involved? It detects what? It detects gravity, So you can say it is involved in gravitropism. Detects, it's the sensor for gravity, gravity sensor. Okay, so that's it. Plant form, that's all there is. Then we can move on to animal form. Right. State the protein carbohydrate complex found in the matrix of cartilage, type of cells that secretes it. Cartilage. Yeah, direct theory. Okay. What is the matrix? If you take any connective tissue, what are the three parts? What are the three parts that you have in any connective tissue? Any connective tissue? You have cells, of course. It's a tissue, right? Cell is the main role. Then the cells usually secrete what? Two things. It usually secretes a matrix, which is like a glue, right? Holding all the things in the tissue together. And of course, there can be fibers, right? If you have a concrete, to that concrete you put steel bars, you put sand, you put chips, kalugal, then you add the cement. It's like the connective tissue is like that. You have several things in the mixture, right? So a connective tissue always has cells, a matrix, and fibers. So if you take cartilage, which is a connective tissue, you of course have a nice matrix, which is chondroitin, chondroitin sulfate. Chondroitin sulfate. And who secretes it? It's a cell. What is the name of the cell? Chondrocytes. So is the word that you use to describe your cartilage. No? Chondro is the word for cartilage. That's why we call cartilage fish as chondric thias. So these are chondrocytes. Cartilage nang chondro. Yes. Bone nang osteo. So many other prefixes are there. Major function of cartilage tissue other than providing support. Cartilage other than providing support. So if you want support, what is your first choice? If you want pure strength, what is your first choice in your body? What tissue? What tissue? Bone. Bone is the strongest. No? If you want just pure strength, you put a bone there, no, to bear up your weight. Yeah. But if you want something additional with that bone doesn't have, right? Something additional that bone doesn't have, you need. If you want flexibility, flexibility. If you want flexibility, you have a cartilage. For example, if you want to support your outer ear, it has to be flexible. No, if it's bony structure, it's all definitely going to get fractured because it's a small thin structure, right? So you want to have a defined shape, but also you want it to be flexible. In the nose also, you want it to have a defined shape, but it has to be flexible because it's an outer protruding structure. Yeah, got it? Flexibility. Then we move to a bit of nutrition. What is known as each of the following? Protein sparing. Protein sparing. Protein sparing means you spare the proteins. You leave the protein alone. Yeah, you leave the protein alone. That happens. I mean, okay. When do you use protein? When you use protein, here what are we talking about? The use of protein to produce energy, right? So in your body, there's a hierarchy of using different substrates to get energy. The most preferred ones are carbohydrates, not carbohydrates. If you have monosaccharides, then good, very easy to produce energy. If you don't have carbs, the next choice is going to be fats. Okay, you can break down the fats into amino acids. Sorry, you can break down the fat into glycerols and fatty acids. Then separate processes are there for the glycerol and fatty acids. Glycerols can enter glycolysis. Fatty acids can enter into the Krebs cycle. Okay? You can extract energy from fats. If you don't have fats also, then you turn to protein. Protein breakdown is a wasteful process because proteins have so many functions, transport, defense, muscle contraction, so many enzymes, all are proteins. So you are going to be breaking down protein as a last resort. Very mono. For example, if you are stranded on in a desert island or something, then after your glycogen stores are over, then your fat stores are going to be over, then your proteins are going to be digested. You are going to lose muscle, you are going to be very thin. So protein sparing is actually not using protein to produce energy, when there is adequate carbohydrate. Not using protein to produce energy when there is adequate carbohydrate. Okay. If you have enough carbs in your diet, you don't need to break down your proteins. Next, non-essential fatty acids. You don't need them essential neve. So they are not essential. That means what? Does it mean you don't need them? No, you don't need to eat them. Non-essential means you don't need to eat them. Then how are you going to get them? You can make them. Okay, you don't need to eat them. You can make them in your body, mainly in the liver. Your liver is like a nice factory. If you give it some raw materials, it can give an output of so many things. Non-essential fatty acids are what? You can say fatty acids synthesized in the body. You don't need to eat them. Similarly, you have non-essential what? Non-essential amino acids are also there. Non-essential amino acids are also there. Out of the 20 amino acids involved in protein production, how many non-essential amino acids are there? Out of the 20 amino acids that you need to make proteins, how many non-essential ones are there? Yeah, 12. Because from 8 that you eat, which are called essential, you can make the remaining 12. Okay, so you need to only eat 8. Okay. Right. Similarly, there are non-essential fatty acids. You don't need to have them in your diet. They can be synthesized in the body. Then there's a simple definition: balanced diet. But there, okay, you can write down. It's a diet containing all essential nutrients. Okay, it contains all essential. It shouldn't be deficient in at least even one. If the diet is not having at least one nutrient that is essential, that is then it's not a balanced diet. All essential nutrients, but something additional should be there in appropriate proportions. Right? Appropriate proportions. Otherwise, it's going to be unbalanced diet. Balance means you should have them in your recommended daily intake. Have you seen RDA? Yeah, recommended daily values. Yeah, in the back of nutrition sort of the label, you must have seen this contains this much of the recommended daily calorie intake, this much of the daily vitamin C, this much of the daily calcium intake, right? They usually try to advertise because if you eat too much of certain nutrients, it could be toxic. For example, certain vitamin A is stored in the liver. It's fat soluble, right? So if you have too much vitamin A, it could be toxic. Got it? So it's like that. If you need to have all your nutrients in appropriate proportions, otherwise it's not a balanced diet. Okay. Two non-essential amino acids. Two non-essential amino acids. What are the two non-essential amino acids? You can mention alanine and cysteine. Okay. Alanine and cysteine. Okay. Do you have any other non-essential in your syllabus? Do you have any other non-essential in your syllabus? What do you think? Did you find any other non-essential in your syllabus? Alanine, cysteine is there. You have, I think, 4 essential amino acids that you need to know, but only 2 non-essential. No? Am I correct? You have alanine, cysteine. Yeah, leucine, methionine. Yeah, only 2 is non-essential is given. You have leucine and methionine as essential as well. Okay, you can write down there. Then so write it down. You have 4 essential in your syllabus either. You have 4 essentials out of the 8. Okay, out of the 8 essential amino acids, you learn about 4. You have leucine, methionine. Write down here if you have the paper. These are essential. These are essential. You have leucine. Then don't you have histidine or something? I think it's given in your, yeah, lysine and histidine also given. These are essential. Huh? You have 4 essential in your syllabus. You have 2 non-essential. So they are the ones that has been questioned. Leucine, methionine, lysine, histidine, those are essential. Okay. Okay, right. Let's come back to the paper. Right. Normal value of each of the following in a healthy adult. Okay, blood pH. Where should you maintain your blood pH? A bit above normal, I mean, a bit above neutral level. A bit above neutral, about 7.4. Yeah, a bit above neutral level, about 7.4. Because of the level of certain basic things in your such as bicarbonates or whatever in your blood, it's slightly basic, very slightly basic, 7.4. Ne? Okay. So this can decrease, become more acidic. This can become more basic. What is the main way of this becoming acidic? Yeah, carbon dioxide is the main one that is going to make your blood acidic. Because why? It's going to dissolve and produce carbonic acid, H2CO3. Then it is going to release H plus, concentration increases, the pH decreases. Lifespan of erythrocytes. Erythrocytes are what? Yeah, red blood cells, right? Quartno samlat exam में केवल तम्मत करो, right? 120 days. Guys, he's just going to live 4 months and will be destroyed. Where is the site of production of red blood cells? Where do you produce red blood cells in your body? Produce bone marrow. Bone. Spleen is a production of place of destruction. You break down medicine after 120 days, they go to the spleen and also the liver to be broken down. Is the recycling plant. Okay. Blood pressure at rest. This is sort of the average value. Yeah. For a normal adult person, into the BP. Above you write the arterial pressure. Then below this arterial pressure is when the heart is contracting. You call that systolic. Below it is called diastolic when the heart is not pumping. The left ventricle is not pumping, but you have to indicate in millimeter mercury. Units, the units don't get cut off. Okay, it's a ratio, but you are indicating both values. It's not a the value doesn't get cancelled off. Okay, you have to write its millimeter mercury. Okay, you are mentioning your systolic and diastolic pressure just one above the other. So it's not going to get cancelled. It's not going to cancel the mercury millimeter mercury unit. Okay. We're moving on to a bit of so we are in circulation. What is known by each of the following? Cardiac cycle. What do you mean by the cardiac cycle? Yeah, it's so if it's a cycle, no, it's a cycle. Now, if it's a cycle, does it have a start or an end? No, it happens again and again, no? Right? So it's a set of events that happens after the other again and again. So you have to say it's a sequence of events. Maybe sequence of events that take place. Study with me, okay? It's a sequence of events that take place in a complete heartbeat. Study with me. It's a sequence of events that take place in a complete heartbeat. What do you mean by a complete heartbeat? Lub dub. Yes. Lub dub sound. You don't need to know what do you mean by the lub sound and the dub sound. Lub sound is actually more loud. Lub sound is heard when the ventricles contract. When the ventricles contract, what's going to happen? Which valves are going to close when the ventricles contract? Blood should not be going back into the atria above. So when your ventricles contract, this is the heart. Okay. When your ventricles contract, which valve should close? The AV valves, bicuspid and the tricuspid should close. Then dub sound is a slightly less one. That is heard when actually the ventricles are relaxing. The ventricles are relaxing. The dub sound is heard when these two vessels, there's a vessel here and there's another vessel that is going home. What do you have here in the opening of these vessels? Semilunar valves. You have semilunar valves. The smaller dub is heard when the semilunar valves close. Okay, so during one complete heartbeat, all those things happen. No? Atria contracts, ventricle contracts, and the heart relaxes. All right, so that's what you have in the cardiac cycle. Next definition, hypertension. Okay, the most important word you cannot miss here, of course, it's elevated blood pressure. No, elevated blood pressure. But is it right now? So, for example, if you ran some 100 meters or something, your blood pressure is going to be elevated. But can you say, do you have hypertension? No, it's a disease. No, hypertension is a disease condition. It has to be a sustained elevated blood pressure. So how do you say it's elevated? You need to know the parameters, no? Sustained elevated blood pressure above normal limits. Blood pressure should be increased, and it should stay increased. Then you will be diagnosed as a person having hypertension. Then you will be given pressure drugs, right? And antihypertensive drugs. Okay. That means a person who's having hypertension, when he's normally resting, he also has high blood pressure, right? That's why we say sustained elevated. Okay, now we're moving on to a bit of respiration, gas exchange. What is known as anatomical dead space? Anatomical dead space. 2 points you have to write here. 2 things you have to write here. If it's dead space, usually means it is not contributing to sort of the living processes. Okay, it is not contributing to living processes. So if you take your respiratory system, it's in your lungs, and also the respiratory tube system is designed to deliver atmospheric air to your gas exchange surface. So what is your gas exchange surface? Millions of sacs called alveoli. So your alveoli are not directly connected to the atmosphere. There's a delivery system to deliver air. Those are just tubes, no? They don't exchange gas. So you have to say the anatomical dead space is the volume of air, you can simply say in the conducting tubes of the respiratory system. Conducting tubes of the respiratory system. Or you can mention, which is the way I like you to write, okay? Volume of air in, what are the conducting tubes that you need to know? You have the main one here, trachea. Then from the trachea, so trachea comes up to sort of here. Then it will divide into go to the two lungs. Bronchi. You have a right bronchus, left bronchus. Together bronchi, plural. Bronchi divides into what within the lungs? Bronchioles. All these are not involving in gas exchange. Volume of air in the conducting tubes, the trachea, so you can call trachea, bronchi, and bronchioles, not contributing or involving in gas exchange. That is dead space. You can add in the alveolus, gas exchange in the alveoli. Okay. What is the volume of the anatomical dead space in a normal healthy adult person? That's important, okay? This set of words, okay? So this is bit additional. Why do we say in a normal healthy adult person? Because due to disease conditions, your anatomical dead space can increase. So in a normal healthy person, the dead space is only these conducting tubes. Imagine you have a disease like silicosis, asbestosis. What's going to happen when you have those diseases? The lung tissue is going to get damaged. Then it is going to be repaired. By what? What by what process? Can you, so imagine due to asbestos particles, part of your lung get damaged. Then can your body repair it exactly as it is? Can you build some new lung tissue there? No, you can't. There won't be any new lung tissue. There would be a damage, but the body will repair it, but it won't be a very good repair job. Can anyone tell how is it repaired? By what process? Fibrosis. Fibrosis. Silicosis, asbestosis, even in tuberculosis, even uh, the lung get damaged and then it is repaired by just fibrous proteins. We call that fibrosis. Okay, these are all in the syllabus. Are the fibrosis? Fibrosis doesn't produce new alveoli. Okay, so it doesn't make the lung better. It's not repairing the lung, just it's a patch job, right? So fibrosis can increase the anatomical dead space. So it's a play, it's part of the lung that is not contributing to gas exchange. No, that's why this word is important. Normal, healthy adult person. In average, it is about, you can mention it in milliliters or cubic centimeters. 150 ml is good. Okay, in a healthy adult that doesn't have any respiratory diseases. State how the coordination. Okay, moving on to nervous. I think I did this comparison when we just started the nervous system. Okay, what are the differences between your nervous system and the endocrine system? Yeah, quickly summarize. What are the differences between nervous system and the endocrine system? What are their strengths and what are their, let's say, in what places do you prefer a nervous conduction? In what places do you prefer uh, endocrine coordination? So we'll quickly remind ourselves of that by the Taggala. Okay. On this side, if you have nervous, if you have this side endocrine, this is, we are talking about the coordination by these two processes. Okay. What are the criteria that you can compare? What are the criteria that you can compare? Basically, how it works. No, you can say transmission. Transmission. So the nervous conduction occurs through what? Through electrical impulses. Neither through, okay, sorry, I mean through neurons, nerve cells. Right? Nervous occurs through nerve cells. Then endocrine, as the name says, I mean, you know, endocrine glands are ductless. So endocrine hormones travel via blood directly. No. Then what is the transmitter? So transmission is this, occurs through neurons. Then what is the transmitter? Who's tracking? What is the medium? How does it travel? If you take your nervous system, it is electrical and mainly electrical, but in some places you can have a bit of chemical. Where do you have chemical? Where do you have a chemical transmitter in your nervous system? Yeah, where do you have anyone online? Where do you have a chemical transmitter in your nervous system? Where? Where do you have a chemical that is going across in the nervous system? Yeah. Synapses. Synapses, you have neurotransmitters. Neurotransmitter is a type of chemical. Neurotransmitters in synapses. In blood, I mean, sorry, in endocrine, it is mainly all chemical. It is all chemical hormones. Hormones can be lipid based, protein based, okay, could be anything. Then, what other things can you consider? The response. This is what I told you. If you are designing a certain system that is either controlled by nervous and endocrine, you have to think, if I want this process to be a very precise thing. For example, me holding my pen is a very precise thing. I require the involvement of some very precise muscles in my hand, right? So for that, I need a sort of a very specific signal to go to a specific place. No, so the best one is a nervous signal because the response, you can say, it is localized. If there is a nerve supplying that particular area, you can send a impulse only to that place, right? But if you release a hormone into blood, it's going to reach everywhere in your body. Okay, so you can say the responses of endocrine hormones are, endocrine system is diffuse. For example, if you have growth hormone in your blood, all the tissues are just going to grow, né? When it's exposed to growth hormone. So this the effect is diffused if you have endocrine. Then Tagala, what else? Time taken to start the response. The neural nervous, of course, very fast, and hormonal coordination is usually slow acting. Okay. Then also the duration of response, it is very short here. Then the nervous endocrine coordination, it's long. How do you remove a nervous or endocrine system signal? How do you stop a nervous signal? Sorry, a hormonal signal. How do you stop a certain signal that is being transported by hormones? You have to remove the hormone, no? The hormonal inactivation happens in what organ? Hormone inactivation usually happens in what organ? So once the hormone has sent its signal, it has to be inactivated. Yeah. Yes. Liver. Right. Liver inactivates many hormones. Okay. Keep that in mind. Those are theory points. One of the functions of the liver, it activates many hormones. Okay. Okay. Taggalena, back to the paper. Right. So you can say state how coordination through the nervous system is faster compared to the endocrine system. What can you say about this? What can you say? Why is it faster? You can say, so you can start with the transmission. Okay, nervous signals occurs through neurons. Occurs through neurons. By what transmitter? What by what transmitter? Electrical impulses, or you can say electrical signal. So that makes it fast. On the other hand, you can mention about the endocrine system. The endocrine system uses chemicals, better to write hormones, okay, better to write hormones that travel via blood. That's why it's slow. The nervous system uses electrical signals that travel via neurons, while the endocrine system uses hormones that travel via blood. Okay, that's why they are faster. Major functional areas, cerebral cortex. If you take this all this convoluted area in your brain, what are the three main things that are done by your cerebral cortex? Everything that is received or sent to the brain will be identified, recognized, and then what do? You will process them, integrate them in a different area. Then finally, if you need to take a step, you need to do something about it, then that action will be taken. So sensory information will be recognized in a sensory area, then it will be processed or integrated in a association area. Then if you need to do something about it by controlling what? Skeletal muscles, you need a motor area. Those are the three parts of the three functional areas of the brain. For example, in the physics paper, you see a question. So using your eyes, you read the letters. That is the sensory area. So you read the letters. Then in the association area, you understand the question. You apply the whatever the theory and the equations that you know. Then you work it out in your thinking cortex. That is in an association area. Then using the motor area, you write out the answer. Okay, They are all controlling all the processes. Differences between sympathetic and parasympathetic division. They are asking differences. Okay, sympathetic and parasympathetic. What do you have to write? Yeah, I think I included a diagram in the nervous system questions. Okay, sorry. Why is it? It's crashed, I think. Hold on. Oh, right. It can't handle much. Okay, right. All right. So sympathetic and parasympathetic divisions, they are asking to do a comparison. Okay, they are asking to do a comparison. Right now, visualize it. Right now, visualize it. Okay. So talking about the sympathetic division first. Okay. What are the two types of autonomic nerves that you have? What are the two types of autonomic nerves based on the place where they exit the spinal cord? Can you see? There's a cervical thoracic lumbar right? So if they exit in this very upper region that we call as the cranial part of the spinal cord, that we call as the brain stem, right? If they exit from the brain stem, okay, they are called as cranial nerves. Okay, then if they exit from the spinal cord itself, they are known as spinal nerves. Okay, so there are two types of autonomic nerves, as you can see here. Can you see? These guys exit from the brain stem. Okay, so if I ask you what are the parts of the brain stem, you can tell me. What are the parts of the brain stem? You have midbrain. Then below that, what are the parts of the brain stem? Pons, varoli, and medulla. So your cranial nerves exit from this brain stem. Spinal nerves exit from the spinal cord. So can you see here the sympathetic division? All the nerves are exiting from the spinal cord. So if you can write in the sympathetic side, nerves exit only from the spinal cord and the, or you can say as spinal nerves. Okay. About sympathetic, you can write nerves exit from the spinal cord, or you can say all nerves are spinal nerves. Or you can say all nerves are spinal nerves on the sympathetic side. You can see that, no? All nerves are spinal nerves. You can write the same thing about the parasympathetic. Can you see? You can write nerves exit from the brain as well as the spinal cord, or you can say nerves, there are cranial nerves as well as spinal nerves. Can you see? There are some nerves exiting from here. On the parasympathetic side, you can write there are cranial nerves as well as spinal nerves. There are cranial nerves as well as spinal nerves. That's one difference. Then you can talk about what they do. Yeah, what do they do? If you come to the sympathetic side, you can say prepares the body for exciting, stressful situations, energy demanding situations, or you can just say fight or flight response. Okay, you can write anything, any of those. You can write, say, prepares the body for energy demanding, stressful, exciting situations, or prepares the body for fight or flight. You can write on the parasympathetic side. Can you see these responses? Promote calming down. You can write promotes calming down, or return to self. Return to self. Okay, or you can say return to normal condition, promotes calming. Yeah, promote calming, return to self maintenance condition. That's good. Yeah, return to self maintenance conditions. If you can write that, that's good. Return to self maintenance. We call that rest and digest. You don't need to write that. Return to self maintenance or promote calming. That is done by parasympathetic. Okay. What else? Then you can talk about the neurotransmitter. If you take parasympathetic, it is acetylcholine. It is acetylcholine. The neurotransmitter is acetylcholine, and on the parasympathetic side, you can write the neurotransmitter is, you can say, noradrenaline. Noradrenaline. What is the other name for noradrenaline? Norepi. Okay. Norepinephrine. Norepinephrine. Noradrenaline or norepinephrine. Those are the neurotransmitters for sympathetic. Okay. Okay. Come to back to the paper. So that's done. So they are only asking two. So you can say what about the cranial and spinal nerve. Then you can talk about their response. Then you can talk about their neurotransmitter. Right. This is for the final time. Study these. This could be the final time that you are getting the chance to study these. Okay. State the disease that causes severe mental deterioration characterized by confusion and memory loss. So think of an old person now. Think of an old person whenever you get these symptoms, right? Severe mental deterioration, confusion, and memory loss. Yeah, happens in what disease? Happens in what disease? Yeah, yeah. It's Alzheimer's, because the word severe, right, refers to Alzheimer's disease. Yes. So Alzheimer, keep it, related to elderly people. You know how elderly people now, they are getting, they don't remember anything. They don't remember where their house is. They get lost, right? They can't, later, they can't dress themselves properly. Okay. They can't do their own work. Okay. It comes under the family of diseases that we call as dementias. Okay. Okay, so it's an umbrella term is there that is called dementia. Under that family of diseases that is called as dementia, you have Alzheimer's. Alzheimer's has a sort of a genetic basis as well. Okay, got it. Keep that in mind. What are the other diseases that you need to know? Nervous psychiatric diseases. Schizophrenia. Schizophrenia, what is characterized by what? You are having an altered perception. You are hallucinating, delusional, right? You are seeing things that are not there, hearing things. Okay, schizophrenia. Got it? Then depression, of course, depressed mood, self-explanatory. Then what is the other one? Parkinson. The Parkinson one is mainly what? It's a motor disorder. You don't have a motor control. Parkinson. Those four have their own unique features. Schizophrenia, you have delusions, hallucinations, altered perception. Depression, you are sad. Then Alzheimer, you forget and confused. Parkinson, you can't control your muscle movements. Okay. Moving on. State an advantage of binocular vision. State an advantage of binocular vision. Why do you need binocular vision? Yeah. Why do you need binocular vision? Yeah. For to judge the depth. Yes. You can say judge distance and speed also. Right. So you can write all these three. You can write 3D vision is one thing you can write. You can get a depth, judging depth, yeah, judging speed, right? All these things, for all those things, you need a two angle, because from your two eyes, you are seeing light is entering in two angles. No? So if an object is moving, as the object is moving, it sends light rays at different angles to the two eyes. So with that, with the association area in your brain, you can process that, and you can get an idea of how fast that object is moving. Is it coming towards you, or is it going away from you? Right? 3D effect, you can say. Function of Eustachian tube. Function of Eustachian tube. Where do you have the Eustachian tube, first of all? Yeah. Where do you have Eustachian tube? So what I like to remember about the Eustachian tube is what situation do you, in real life, you think about your Eustachian tube? Whenever you have are locked ears, right? If you are in a plane, or you are sort of climbing a mountain in a vehicle or something, you get your ears locked. That happens because if you take, this is the external auditory canal and this is the middle ear. Okay, this is the middle ear, and then you have the cochlea and the three semicircular canals here. Then there's a tube that is connecting. So this is the ear. Okay, right. There's a tube that is connecting this is your middle ear. No? Middle here you have the three ossicles here, right? There's a tube that is connecting this middle ear to where? To the pharynx. Here you have the pharynx, throat. Okay. When, so in your middle ear, there's a membrane, right? Quickly, this is quick revision. There's a membrane here. What do you call that membrane? From the external ear. Where is the external ear end? At the cumbere, eardrum. Tympanic membrane. Method tympanic membrane is there. Then there's another membrane here that is called the oval window. So between these two, you need to maintain atmospheric pressure. Okay, you need to maintain atmospheric pressure. So the only way to do that is through your mouth. Yeah, through your mouth you can maintain atmospheric pressure. Okay, so as you are in a plane or going up a mountain, the pressure is going to drop, but it is not going to immediately equalize. That's why we feel like your tympanic membrane is going to start to hurt, because why? The inner pressure is actually higher than the outside, because you are going into a less pressure area. So what do you need to do? You either swallow a bit of saliva or chew some gum, so that more air goes into your mouth and goes into the middle ear and equalizes things. Okay, so you have to say what is the function? Maintenance of air pressure on both sides of the tympanic membrane at the same level, or you can say atmospheric level. So what are there's only one point there. Maintain air pressure, maintain air pressure on both sides of what? Tympanic membrane. Both sides of tympanic membrane. It could be at same level, or you can say at atmospheric level, no? At atmosphere. So that's why it's kind of painful, no? We can ears getting locked. It's kind of painful. That is because the tympanic membrane is hurting. Your tympanic membrane is getting hurt because of the differences in the pressure. Okay, that took a lot of time to discuss just one structured essay question. Okay, right. We'll directly go into the next question. So there's going to be some reproduction and some support. No, immunity. Ah, yeah. It's a bit of a mix. Yeah. Let's see. Question number 3. Name a phylum of animals that contain a hydrostatic skeleton. Hydrostatic skeleton. Where do you have a hydrostatic skeleton? You have either two options you can write. You can write two options. Who's the first one to develop a hydrostatic skeleton? Evolutionarily, Mulimba, which phylum? Nematodes. Yeah. Nematodes started to have the first skeleton, hydrostatic skeleton. But so hydrostatic means there's a fluid trapped between two layers of muscle, body layers. Nematodes, what acted as the hydrostatic skeleton? Pseudocoelom. Pseudocoelom. Because they didn't have a proper coelom yet. They are pseudocoelomic. They are pseudocoelomic. So they didn't have a proper coelom yet. But their pseudocoelom acted as a hydrostatic skeleton. What is the other one? So the other phylum that you can write is Annelida. So in them, you had the coelom, proper coelom they had. Filled with fluid. So it's like a pressurized fluid. So it can give you some support. State one function of each of the following in the human skull. Fontanelles. So when you were a baby, you had fontanelles. Okay. When you were born, it helped your mother. If you are a normal, you were born normal vaginally, not a C-section. Okay. You can say allows compression of the skull. Allows compression of skull to facilitate parturition. To facilitate parturition. At birth can give mark, but prefer write the posh word. Okay, parturition. At parturition. Allow compression of the skull, facilitating parturition. Facilitating parturition. So the baby is much easier to travel through the vagina and come outside. Sutures. Again, in the skull, why do you have sutures? So you actually, the fontanelle areas will later fuse with sutures. Okay. So sutures actually, there's no actual function. Sutures actually, you can say, connect cranial bones tightly. Cranial bones tightly or make cranial bones immobile. Otherwise, it's not going to be a very sort of a helmet-like structure. You have the cranium. So a cranium is a sort of a firm cover. Connect cranial bones or make the cranial bones immobile. Okay. Which human vertebrae contain a foramen in each transverse processes? In each transverse process, which type of vertebrae you have a foramen? What do you mean by foramen? An opening, no? Which type of vertebrae has a foramen? Cervical. Why do you need a foramen in the cervical? Why do you need a foramen in the cervical? I think I showed you. Why do you need a foramen in the cervical? Why do you need a foramen? Yeah. A foramen is there and it's an opening. No? An opening is usually there to allow someone to go. Who is the someone that is going? LM people you can see, right? If you zoom into this guy's skeleton, maybe these are the cervical vertebrae. Can you see the cervical foramens? These are the foramens in the lateral process of cervical vertebrae. So if I add the blood circulatory system, you can see who's going through them. Hmm. There's someone going through them. Ne? Maybe who is that? Vertebral artery. That is called the vertebral artery. Okay, this one, vertebral artery. Okay, to allow the passage of the vertebral artery, you have the cervical. In the lateral process, you have for this is the carotid. So this is the carotid artery, common carotid. It has two parts as the internal and external. Okay, right. The carotid artery is important because why? You have something in your syllabus from the carotid arteries, particularly Menometan here. There's a sort of a expanded part here. Yeah. That is called the carotid sinus. Method again, a carotid sinus. Do you have something in the carotid sinus? Yeah. There's a chemoreceptor in the carotid sinus that can detect carbon dioxide, not I mean the pH change. In here, Metin, this is called the carotid sinus. In the carotid sinus, you have a chemoreceptor that can detect the pH. Yeah, involved in regulating respiration. Okay, got it? Online people clear, right? Okay, so they're clear about the foramens. Okay, vertebral foramen. What else? Two example for hinge joint in the found in the human lower limb. Hinge joint found in the human lower limb. Where do you have hinge joints? A hinge. A hinge means what? It's like a door. You can only what extend and flex. Extend and flex. In the lower limb, where do you have? You have the knee joint. Where else do you have a ankle? Yeah. Those are the you can that's the thing with bio. You can just do in your body and see if it's a hinge joint. Can you only extend and flex? No, then it's a hinge joint. Like your elbow. Your elbow is a very ideal hinge joint. Okay. In the knee joint, what is the other additional thing you can do? In the knee joint, there's some special point. Okay, I'll give you a hint. It helps you to stand. Yeah. If in the knee joint, there's another special movement that you need to know. That's going to help you. Okay. So, if you're standing on a school assembly, if you have been standing in a school assembly for a long time, there's a mechanism in your knee that is going to help you stand for a long time. That is called the locking mechanism of the knee, right? Some people have this hyperextension, right? So their knees go slightly behemoth, right? Go backwards, okay? But when the knee is locked, when we say that the knee is locked, the weight of the entire body sort of shifts from your muscles into the bone so that the bones are nicely stacked on top of each other. Then you can stand for a long time. That is called the locking mechanism of the knee. Locking mechanism, okay, that allows you to stand for a long time. So your knee can flex and extend, of course, and then there's a locking mechanism. It's a rotating motion. Okay. Okay, enough. Okay, we'll come back to the paper. Name a group of animals which possess salt glands. Salt glands. Ah, yes, something I missed here. Somewhere else that you can have hinge joints similar to your fingers. Your fingers you can only flex and extend. Toes. Toes. So you can't just say toes, right? You have to say joint between phalanges. Phalanges. Phalanges are what? Toe bones, finger bones are both called phalanges. Ne? Yeah. Knee joint, ankle joint, joint between phalanges. You can bend them, no? Bend and extend, bend and extend. Group of animals which possess salt glands. Salt glands. Salt glands are actually found in your skin. Marine birds and marine reptiles. So they actually have in your marine birds, there's a gland sort of located behind their eyes, and it sort of secretes salt along their beak. Salt is secreted along their beak. Okay. Marine birds, certain marine reptiles are also having salt glands. Marine birds and marine reptiles. Okay. So excretion, some question is there. Name two substances that are secreted by the distal convoluted tubule. Secreted means from blood into urine, right? The secretion happens because why? In your glomerulus, these things were not properly filtered. So later in your nephron, they will be sort of especially secreted because they didn't get filtered. They escape secretion, sorry, escape filtration in the glomerulus. So now what you do is later in the nephron, you somehow put them into the urine. I mean, you forgot to filter out these things. So I'm going to just secrete them. What are the things that are secreted? Things actually in the distal conductible, we can actually control these. The two things that are secreted are H plus and potassium. Can anyone mention what is the hormone that can control how much of these are secreted? And okay, then hormone and the gland. H plus and K plus secretion in the distal conductible is under the control of hormone. Anyone? Aldosterone, which is a, what are these? Potassium is a mineral, mineral. Then you can call it a mineralocorticoid. So the name has a corticoid. Corticoid means it is secreted by a certain cortex. So who has a cortex? Adrenal gland. So mineralocorticoid. So aldosterone is a mineralocorticoid secreted by outer adrenal cortex. Neither. Take it down, right? Quickly write it down. It is under the control of aldosterone hormone, which is a mineralocorticoid secreted by outer cortex of the adrenal gland. Okay. Okay. Shall we move on? That also reabsorb sodium and water. Sodium and water reabsorption and H plus K plus secretion can be controlled. Yes. Yeah. Yeah. So we are talking about here secretion. So I didn't mention those. Okay. Two sides of action of ADH. What is the long name of ADH? What is the long name of ADH? Anti You can write here, it is antidiuretic hormone. Diuresis means you are losing water. Right? The diuretic hormone. So antidiuretic means you are doing opposite of losing water. That means you are saving water in the body. Ne? So where does the ADH act? It recovers water from distal convoluted tubule. You write it properly here, the distal convoluted tubule, and also collecting duct. From the distal convoluted tubule and the collecting duct, it reabsorbs water. Okay. Then the next question is a bit of a rare question that has been asked in this year's paper, that I mean 2021 paper. Okay, I will show you, explain that using a small diagram. Hold on. That is about helper T cells. Okay, helper T cells. I mean, it's a bit of a hidden point that they have asked. Okay. Okay, check this diagram. Listen carefully. Okay. So here you can see this. This is called a helper T cell. Okay. So this is actually an antigen presenting cell. Antigen presenting cells, you know, because if you take T lymphocytes, lymphocytes are very picky, no, because they are not going to respond themselves, and they are going to, they have some sort of a, I mean, if you have a certain pathogen in your body, T lymphocytes are sort of, they are sort of a, their ego is too high, so they themselves are not going to recognize. They want some servant to come and show them, okay, there's a pathogen here, right? That sort of cells are called antigen presenting cells. So this is a dendritic cell that is acting as antigen presenting cells. Can you see? Ignore all the other things that you don't know. Okay. So this pathogen will be engulfed by a antigen presenting cell. It could be a macrophage. You know the list of antigen presenting cells. Macrophages, dendritic cells. Right. Then can you see that pathogen will be broken down into certain antigen pieces. Then it will be presented. Okay. Presented, and that presentation is can be to a helper T cell. Okay. That helper T cell will help to then go and activate. Can you see? It will go and help. So now this helper T cell is activated. Okay. The helper T cell has the additional effect of secreting cytokines as well. Cytokines are what? Sort of immune signaling molecules, right? So these cytokines sort of activate more helper T cells, and also the helper T cells proliferate. Now you have a clone of helper T cells. The helper T cells will help both the B cell mediated humoral immunity as well as the T cell mediated uh, cell mediated immunity as well. So both will be helped by helper T cells. So as the name says, they are sort of in the background facilitating both the processes. Okay. So you can write down the functions of the helper T cells. Okay, you can write down the functions of helper T cells. The final function is enough. They help to activate both T and B cells. So you can write activate or provide signals to activate cytotoxic T cells. Cytotoxic T cells go what? They kill cells, no? They kill infected cells. Cytotoxic T cells kill infected cells. Okay. Then what is the other thing? So that is the T immunity. No? Then activate B lymphocytes. They secrete what? Antibodies. Those are the functions of yeah, T cells, helper T cells. It's a bit of a hidden theory point, but they have asked this. Okay. Okay, moving on. What is the reason for developing type 1 diabetes? Type 1 we call as immune mediated diabetes. Okay. Immune mediated, this is usually found in small children, right? Babies can be born with type 1 diabetes and it can develop in childhood, type 1 diabetes. It is, can you give me another name for this type 1 diabetes? Yes. Type 1 diabetic people, okay, I'll give you the answer, right? What's going to happen? The beta cells. So, okay, you can work this out, okay? So, you have diabetes means you have uncontrolled glucose in your blood, ne? What is one of the things that can cause uncontrolled glucose levels is that you don't have enough insulin, ne? Yeah. So, if you don't have enough insulin, then it could be a problem in the person developing insulin, ne? Producing insulin. Who's producing insulin? Beta cells in islets of Langerhans, where in pancreas, ne? Okay, so now apply your knowledge of immune cells. These are beta cells. So, who is able to damage another cell? Who is able to kill another cell? It is a cell able to kill another cell. Who is the only cell that is able to kill cells? T cells. T cells are able to kill other cells. They are like people with guns. They can go and kill another person. A person having a gun can kill another person, right? A T cell is like a cell killing cell. Okay. So beta cells will be damaged or destroyed by T cells. You can say cytotoxic T cells. That's what happens in type 1 diabetes. So they don't have enough insulin. So what is the other name that you can give to this type 1 diabetes? Yeah. What is the other name you can give for type 1 diabetes? If they don't have insulin, what do they have to do? They have to take in insulin, no? So it is insulin dependent. It is insulin dependent diabetes. Okay, there's a nice question here, right? So these kind of questions are good because I mean, it's they ask these questions in a fresh way, not just writing. Because this is one of the, I mean, you study it in this way, right? You draw flow charts and you draw these sort of arrow diagrams. Those kind of things you draw to study by yourself also. So when they ask something like that in the exam, it's a bit refreshing, yeah. So you can just use the space and sort of nicely in a free hand you can draw it. So what are they asking? Action of oxytocin on the mammary glands. Okay, on the mammary glands, how many hormones are acting? There are two hormones acting on the mammary gland. One hormone is, what are the functions of mammary gland? Production of milk, secretion of milk, or release of milk. Who is the hormone that is stimulating production of milk? Prolactin. Then the other function is what? Milk ejection, no milk release when the baby wants the milk. That is done by oxytocin. Right. So this is a cyclic process. Now the mother, the female has a baby. So when you have a baby, you need milk. No? So what is the baby going to do? Babies as the fresh, I mean newborn babies, they have only a few um, things in their mind, hunger and sort of other things, mainly hunger, right? So they are sort of programmed when they get near the breast to sort of connect into it. Latching here, they latch into the breast and they start to do what? They start to what we call as suckling. Ne? They start to suckle. So we'll start there. Yeah. So the baby starts to suckling by the baby. Suckling will do what? You can add a step here saying it will stimulate the touch receptors in the nipples. Okay. So touch receptors in nipples. The mother's areola sort of around the nipple, you have some touch receptor sensitive. No? That will send signals to where? Where do you have our oxytocin hormone? It is actually produced by hypothalamus, but stored where? Stored in posterior pituitary. I have always mentioned like posterior pituitary is like the back, the garage or like the back sort of behind your house. You can have a storage area, right? If the so the when you don't have enough space in your house, you put them in a sort of a shed outside your house. You know? So when the hypothalamus doesn't have space, what did it do? It produced the posterior pituitary so that it can store all the excess things. So hypothalamus will produce oxytocin. It will store it in posterior pituitary. Then posterior pituitary will do what? Release oxytocin. It's a simple flow chart. Don't be scared of this. Right? Oxytocin release. Oxytocin comes and acts on the mammary glands. Yes, simple thing, right? Hypothalamus, and it is stored in posterior pituitary. That's all you need to remember, right? The oxytocin will go flow, travel through the blood. Will come to the mammary glands. In the mammary glands, what's going to happen? You release milk. Milk is released because of some smooth muscle contract, right? In the milk ducts. Okay. Then you have milk availability. So better to write. This is a positive feedback loop. So when the baby gets more milk, he will start to suckle more, right? Because milk flavor again, milk is coming out, so we'll start to suckle more. Then there will be more touch receptors, more oxytocin, more milk release. This will continue until what happens? Continue until what happens? The baby stops suckling, because when is the baby going to stop suckling? When its stomach is full, but is going to stop suckling. Okay, so the mother is usually mother is designed to sort of fulfill the needs of the baby completely. Okay, the mother cannot control her own milk release. It's in the control of the baby. Okay. Right. State two advantages of asexual reproduction. Right. Read the question very carefully. Carefully. Okay. Usually, asexual reproduction has. I mean, evolutionarily, it's not very advantageous. Ne? We talk a lot of about the disadvantages of asexual reproduction. So read the question very carefully. Okay. They are asking advantages. What are the things you can get by doing asexual reproduction? Okay. So, it is based on mitosis, no? So, mitosis means you don't need two people to get involved, right? Paternal and maternal organisms are not required. Therefore, you can write. I'm not going to write, okay? You can write only one parent is required. Yeah? Only one parent is required. Usually, because it's mitotic division, allows rapid multiplication for individuals. Then, one of the main things, no energy spent in finding a mate. Yeah, you can understand the human population spends a lot of energy finding a mate. Yeah, right? Maybe that is evolution going backwards. Okay, right? No time or energy spent on finding a mate. Then sometimes this could be an advantage, this could be a disadvantage. So better to write only the above things, but the things after this could be a disadvantage. You can also write genetically identical offspring is produced. Okay, genetically identical offspring is produced. Can you understand? It could be an advantage as well as a disadvantage. Because if you are producing genetically identical organisms, what is the biggest disadvantage? There is no evolution. There is no evolution. There is no change in composition so that the next generation is better suited. Yeah. So you can say, you can write, then you can write another thing. The offspring is genetically identical to the parent. That could be an advantage if what? If the parent is nicely, if the parent is already living in an ideal environment, right? But if the habitat or the environmental conditions change, this could be a disadvantage because the offspring cannot evolve. Yeah, got it. So better write all the first three. One parent is required, allows rapid multiplication, and no time or energy is needed to is spent on finding a mate for reproduction. Okay, let's go. Correct sequence the entire process of production sperm in a man starting from spermatogonial stem cells. Okay. So from the stem cells, so we don't need to start from the embryonic stem cells, but we are going to go from the spermatogonial stem cells. So I think last week I talked about embryonic stem cells. Embryonic stem cells have the ability to become any cell in your body. They are the cells found in the blastocyst, the inner cell mass of the blastocyst. Okay, so no need to start from the embryonic stem cells. After they have differentiated into the small babies, small testes, you can start from there. You can start from, they have given, no? Right? I'll start from there. I don't need to write that. From the spermatogonial stem cells, what are you going to have? It's going to divide by mitosis. Yeah, still mitosis. Huh? You have to mention what is the cell division. You're going to get spermatogonia. Ethylene, mitotic. Then there's going to be one more mitotic division until you get primary, yeah, spermatocytes. Primary Then there the mitosis is going to stop. Mitosis is going to stop. Then onwards, it's going to be meiosis. Primary spermatocytes divide into secondary spermatocytes by secondary spermatocytes by can. What is cell division? Meiosis. Actually, meiosis 1. By meiosis 1. Then the secondary spermatocytes are going to divide into spermatids by meiosis 2. Okay. Follow these in the resource book as you are going through, as we are going through this. Okay. Follow them in the resource. Okay. Then spermatids are of course going to differentiate into our final step, that is spermatozoa, or you can say sperms. Spermatids are differentiate into spermatozoa or sperms. Okay, take this down. Okay, right. From which portion of the blastocyst? Okay, does the fetal portion of the placenta develop? I think when I'm explaining embryonic stem cells, I talked about it. What are the two parts of the blastocyst? Taggal again. You have, yeah, trophoblast, which is the answer for this, right? You have the trophoblast, which is the answer for this. Okay, then the other part that is going to be the actual baby is called inner cell mass. The inner cell mass. Okay, clear? Neda? Your blastocyst has two parts, as the trophoblast and the inner cell mass. From the trophoblast, you are going to get the embryonic, from the baby's side of the placenta is going to come from the trophoblast. Trophoblast will develop into the fetal portion of the placenta. Yeah, and the inner cell mass is going to be the actual baby. So, where do you get your embryonic stem cells that we did last week in applied biology? From here, from the inner cell mass. Because this ball of cells is going to become the entire baby for all the tissue systems, nervous system, bones, muscles, digestive epithelia, skin, everything is going to come from this inner cell mass. Okay. Indicate below how the levels of ovarian hormones in the blood are changed during typical 28 reproductive cycle. Typical 28-day reproductive cycle of a mature woman. Okay. What are the key landmarks that you need to know? If you are given a 28-day cycle, so what are the key landmarks? Which day is important? Day 14. Yeah. Reproduction is good. Right? Right. I think it was, we covered it thoroughly. Ne? Mid cycle, day 14, you have to first of all better to take a dotted line. That is going to help you to draw easier, to make it easier for you to draw. Then you need to identify what are the hormones you need to draw. They are asking ovarian hormones, ne? Hormones produced and secreted by the ovary. Yeah, so they are going to be estradiol and progesterone. What are the other two hormones? You have FSH and LH. FSH and LH are secreted by who? Anterior pituitary, right? Anterior pituitary. So anyway, so you need your ovarian hormone levels. So which one do you draw first? I recommend that you draw your estradiol levels first, because estradiol is secreted by your developing follicle, right? But in the very beginning, because the follicles are small, estradiol levels are small. But as the follicles start to get big, the estradiol levels reach a maximum value kind of 2 days before, two to almost roughly 2 or 3 days before day 14. What happens in day 14? What happens in day 14? Yeah, what happens in day 14? Usually ovulation, no? Mid cycle ovulation happens to. So you have your estradiol levels, then so 2 to 3 days before the ovulation, your estradiol levels reach their peak value. Then they will be maintained by who's going to be here after ovulation. What do you call that phase in the ovary? This part is the follicles are developing, follicular phase. Afterwards, luteal. Ne? I'm talking about ovarian phases. So in the luteal phase, who do you have? Corpus luteum. Corpus luteum does what? Secrete estradiol and progesterone. So they will remain at a sort of a slightly elevated level, of course, higher than this. Got it? So that is the level of estradiol. Then progesterone doesn't have a bit of a, it doesn't increase much during the follicular phase. So it sort of stays at a low, very low level, very low level. It will only increase after the corpus luteum is formed. It will only increase after the corpus luteum is formed. So you have to, so progesterone, this is progesterone, is mainly secreted by your corpus luteum. Therefore, they are compared to your estradiol. Estradiol is secreted by here. Who's secreting estradiol here in this? That is a developing follicle. Developing follicle is secreting estradiol in this before the ovulation or the day 14. Okay. State the actions of the Depo-Provera injection in human females. Something again, something a bit hidden, right? Depo-Provera. It's a type of. Why do you get Depo-Provera? Is it a vitamin injection? Yeah, it's a type of contraception. Depo-Provera, uh, why do you give it? What is the composition of Depo-Provera? It's actually high concentration progesterone, right? Concentrated progesterone injection, right? Main thing is, so when you have the cervix, this is the uterus, this is the vagina, right? Here you have the opening of the uterus, that is the cervix. When you have high levels of progesterone, it is going to have an action that we call as thickening of cervical mucus. Thickening of cervical mucus. Okay. So that sort of prevents sperms that are coming this way can't go into the uterus. Got it? Thicken cervical mucus, preventing sperm entry. That's a first proper answer. Okay. Thicken cervical mucus, preventing sperm entry. Then the other thing, when you have just progesterone, this one has just progesterone. The other thing that's going to do is it is going to make this inner lining of the uterus is called what? Endometrium thin. When you don't have a very thick, rich endometrium, the implantation doesn't happen. Okay, makes endometrium thin. This is going to affect what? You can say preventing implantation. I think I showed you a Depo-Provera. It's an injection bottle. Okay. Depo-Provera you need to get every 6 months or something. No, wait. Every exactly can't remember how. What is the duration? Depo-Provera. Depo-Provera. It has some side effects that so that many, not many. I mean, not big side effects. There's some amount of weight gain, other things. Okay, so some many females don't like it at a bit, right? Yeah, it's a progesterone only. Yeah, every 3 months you need to get the injection if you are interested. Okay, I want every 3 months you need to get effective 99%, more than 99% effective if you get on time injections every 3 months. Yeah, you can have weight gain, some headaches, mood changes, acne, pimples can be there because these are abnormal hormone levels are going to be there. Okay. Moving on to some microbiology, right? What are known as microaerophilic organisms? I think we had this in our microbiology SCQ discussion. We did this. What are microaerophilic? I took these questions. I'll quickly give you the answer so that what do you mean by microaerophilic? They don't like atmospheric oxygen, but they are not completely anaerobic as well. They're sort of in the middle. If you take like a scale, people who don't like oxygen at all, they are anaerobic. Then people who don't like, who want oxygen at all, always they are obligate aerobic or you can say aerobic. Then there are people who are in the middle, they want oxygen, but not as much as atmospheric concentration. So you can say organisms that grow only in low oxygen or oxygen levels lower than air. Oxygen levels lower than air. Then you can write an example as lactobacillus species. Then heterocyst have thick walls. Heterocyst has a precious content inside. Can anyone tell me what does heterocyst have? Enzyme called nitrogenase. Nitrogenase enzyme is found in heterocyst. What is the main problem with nitrogenase enzyme? If you get it into contact with oxygen, it is going to be inactivated. Okay, so you have to protect nitrogenase from oxygen. Therefore, you need to have a thick wall preventing oxygen from diffusing in. Okay, Others, can you hear me, online people? Yeah. The I hope you're not asleep. Yeah. Then dry heat, where use of sterilization of materials in a microbiological laboratory. Okay, I told you the best way to sterilize if you are using heat is what? Moist heat, autoclave, because steam has a lot of benefits. I mean, it's the steam is very effective. Okay, but there are instances where you can't sterilize using moist heat. Then you can use dry heat. Two methods where dry heat is used: you can direct flame, that means you can directly hold it to a Bunsen burner flame or something. Then there is a hot air sterilization happening in a hot air oven. Okay, then there's the third one, uh, which is not going to be done in a, can you understand, microbiological lab. What is the third one that is where you use dry heat? What is the third type of dry heat sterilization? Okay, you have direct flaming, hot air oven. You can just burn them in an incinerator. Yeah, you can incinerate them. No, incinerate. There's an incinerator ovens are there. Incineration is not done in a microbiological lab, no, because incineration is mainly separate used for hospital waste. Okay, hospital or contaminated waste. Yeah. Drinking water treatment, disinfection used in drinking water treatment. Yes. What is the thing everybody knows? Chlorination. In other countries, in Sri Lanka also, some of these, I think household filters have this technique. Ozone filters, I think they advertise some of these water filter brands. Okay, third one, ozone. Sorry, second method, ozone. Name a fungal species and a bacterial species that can cause food intoxication. Right? What are the two types of diseases or problems that you can get due to ingesting spoiled food? Yes. What are the two problems? What are the two types of problems you can get if you ingest food with microorganisms, or you can say spoiled food? Yeah. What can happen? What can happen? What are the two problems? So they are asking food intoxication. What is the other name for food intoxication? Intoxication means it's poisonous. So you can say food poisoning is food intoxication. Eh? Food poisoning. What is the other thing? From the food, you can get an infection. So, foodborne infection, no? Foodborne infection or food intoxication. This depends on the speed of the action. Okay, in both situations, you have microorganisms in your food. Okay, in foodborne infection, so you eat the microorganisms, they multiply in your body, and then they produce toxins and they damage your cells. For example, when you have dysentery, they damage your digestive system cells, so you get a massive diarrhea with blood and other things. Okay, but in food intoxication or food poisoning, the bacteria has multiplied a lot in the food, so there's already a lot of toxins in the food. So as soon as you eat it, within a few minutes, you are going to get the symptoms. Okay, food poison. So now they are asking food intoxication or food poisoning. Fungal aflatoxin, right? Aspergillus flavus. Flavus, scientific name. Aspergillus flavus. Bacterial species, you have two options. Neither you can say Staph aureus or Clostridium botulinum. Staph aureus or Clostridium botulinum. Both of those can cause food poisoning or food intoxication. Okay, right. Few questions. State two differences between subunit vaccines and live attenuated. Again, in micro, did we do this question? Subunit versus live attenuated? Live attenuated vaccines are a bit dangerous, ne? Because of this word, ne? The man or the enemy is still living in the vaccine, yeah? So, when you give them, you are going to get some symptoms, right? Some percentage of the disease you are going to get. Okay, that's why if you get live attenuated vaccines, you can get a fever, body aches, right? Subunit vaccines, they don't contain the even not even the full person. So in live attenuated, we have the living bacteria, but it has been weakened, so it can't multiply or it can't produce toxins or whatever. Okay. In subunit vaccines, you only contain the antigenic portion, so that you are going to develop an immune response, but not a disease at all. Okay. So you can say subunit vaccines contain antigenic fragments, Then, because they are sort of inducing a weaker immune response, you need booster doses. Okay, you are going to need booster or repeated vaccination. You are going to need booster doses or repeated vaccination. Then, on the live attenuated side, you can write contains living, okay, living but virulence reduced, or you can say weakened, pathogenicity reduced, anything you can write. Okay, you can say they contain live or microorganisms whose virulence is controlled, or you can say weakened, pathogenicity decreased. Then the other thing you can say does not require booster doses. Lifelong immunity also you can write. Instead of this, you can write lifelong immunity. Okay. Production of vinegar using fruit juice and name one species. Okay. Vinegar is kind of not very commonly produced in Sri Lanka using fruit juice. We use making what? Usually from sort of the Palmyra sap, no? Right? From the, sorry, coconut sap. Okay. So what do? You do alcoholic fermentation. You produce first ethanol, ne? You produce first ethanol using what? Your friend, no? S. cerevisiae. Write the first proper name, cerevisiae, Saccharomyces cerevisiae. Okay. Then you do acetic acid fermentation for the ethanol. What are the two steps? Alcoholic fermentation, acetic acid fermentation. Acetic acid fermentation, you can do it by acetobacter or gluconobacter. Acetobacter or gluconobacter. Okay. Alcoholic fermentation, what are the steps? You convert the sugar in your fruit juice into ethanol neda alcoholic fermentation done by Saccharomyces cerevisiae. Then in the acetic acid fermentation you convert the ethanol to vinegar, yeah, which is acetic acid. Acetic acid. Okay. Right. Shall we move on to question number 4? Right. We'll see how much we can complete within the day. Because the final question is going to be a nice amalgam between what? Molecular, genetics, and some environmental and applied as well. Okay. Two types of signals that are responsible for epigenetics. What do you mean by epigenetics? First of all, epi means things that are outside genetics. Things that are outside genetics. That means when a person's phenotype is being, phenotype means how he appears, is being determined by factors that are not under genetic control. Okay, determined by factors that are not under genetic control. So it could be certain inherited signals. Inherited signals means sometimes this can include methylation and demethylation. What do you mean by methylation and demethylation is sometimes a gene can be, so imagine there's a nucleotide sequence. A gene can be hidden by a methyl group. When you have a methyl group bonded to a gene, the DNA, what is the first step of protein synthesis? Transcription. You can't copy the information into an RNA, no? Yeah. So when there is some inherited signal sort of masking a gene, we call it a gene is masked, sort of hidden. When that happens, that gene will not be expressed. In the other thing, the other way can happen also. This methylated group can be removed suddenly, so that the gene becomes active again. Both of those can affect a person's appearance, phenotype, yeah, external characteristics. Inherited signals are there, then environmental factors, very important. Or you can say environmental signals. That is because the person's climate, the person's the country he's living in, the climate, what kind of food he gets to eat. Yeah, if he has to work hard a lot, or if he's a very lazy person who is just lying there, then all those things are going to affect how the person looks like, right? The phenotype, the nutrition, the environment, the weather. Okay. Signal peptide present in certain polypeptides. Now, if you are listening to genetics, sorry, this is from molecular. I'm sorry, this is from molecular. Comes under what topic? Signal peptide. What is the example I used to explain a signal peptide? Yeah, if you were listening, in this, when I was explaining the fate of proteins, okay, the fate of proteins, the example I used was if you come to a hospital at the OPD, imagine you meet a doctor at the OPD, they say you need to get an X-ray. Then they hand you a small piece of paper or a sort of a chit telling you to go to the X-ray department. So that you take that chit and go to the X-ray department and get an X-ray done. Or they say to give a blood so that you can we can do a blood test. So you go to the hematology department to give a blood. Like that, you will be guided to different locations in the cell in the hospital by a piece of paper like that. So if you are a newly synthesized polypeptide chain, you were just synthesized, you came out of the ribosome, then you need to undergo some modifications. So to undergo those modifications, there's a piece of polypeptide, I mean a segment of polypeptide that is going to guide you to particular locations in the cell. That is what we call as signal peptide. So you can write the signal peptide guides the polypeptide to particular locations in the cell, or so it can guide to certain locations, so you can the peptide can go out of the cell or to be secreted. It can be packed into a vesicle and could be sent out. International as a sort of international mail with it, you can go out of the cell. Right? It can be sent out. Okay. Okay, who is this? Identify. Name the parts. Identify the molecule. Yeah, this is tRNA. The man is transfer RNA or tRNA. So tRNA is the helper for protein making. Is the helper for protein making, right? So, so imagine you are helping your mother to make a cake. You need to bring certain things, no eggs, flour, sugar, everything. You need to go and give it to her. Bring and go, give it to her. So similarly, in this tRNA, there's a place for the what? What is it? This is called the P arm, where this is the place where the amino acid binds. So you can say site where amino acid binds. That is the P arm. Then Q. Who is Q? Imagine you have a mRNA here. Imagine you have an mRNA molecule here. Then in this mRNA, there's a part 3 nucleotide section that we call as codon. This Q is matching to that codon in mRNA. So එයාට කියනවා මොකද්ද? codon ගේ opposite එක is the anticodon. Anticodon. What is the property of the genetic code that allows a gene isolated from an organism expressing the same polypeptide chain when inserted into another organism? Yeah. So genetic code is almost universal. You know, it's universal. So when you have a certain nucleotide sequence in your DNA, if you can insert it into another organism, it will give out the same result output in that organism. Okay. So that's why we say the genetic code is universal. Right? That's the answer. Right? That allows a gene isolated from one organism in expressing the same polypeptide when inserted. Yes. So what you can say, the genetic code is universal. Universality. Genetic code is universal. That's why one of your genes, when it is inserted into the E. coli, it will produce what? Insulin. Yes, two methods that can be used to introduce a foreign DNA into a plant cell. So when it's given as plant cell, one unique method comes to my mind. What is a sort of the very special method that you have, specialized method, using a special bacteria that can cause a tumor on the plant? What is that bacteria? Agrobacterium mediated transfer is there. So best to write that one first. Yes. You call it agrobacterium is the species. I think, I hope you can remember that this agrobacterium normally causes a small tumor on the plant, right? That tumor is caused because the part of the agrobacterium is transferred into the plant cells. So using that normal mechanism of the agrobacterium, you can use it to put a piece of DNA that you want by modifying the agrobacterium. So you can say agrobacterium mediated gene transfer. That is one good way. Or you can use a plant virus vector. There's a special name for this. It's very good if you have written this. What is the name that you give to virus? Using a virus when you are, because you know, right? Virus likes to integrate DNA into the host cell. That is called transduction. Okay, transduction. You can write that down. Then there are posh things such as the gene gun. Yeah, you can do. If you can't remember anything, just write gene gun. Yes, any method. If you can't remember agrobacterium, blah blah. If you can't remember, write gene gun. Very rare, very rarely done. Okay. Then finally, you can just use a plasmid vector. No? You can use a plasmid vector. What do you mean by a plasmid? Oh, wait, plant cell. Yeah, you don't have plasmids in plants. Ne? You can't use a plasmid vector. So, yeah, you can use a gene gun, agrobacterium, or a virus, a plant virus. Why can't we use plasmids? Why can't we use a plasmid? Plasmids are only found in usually prokaryotic cells, no? Bacterial cells, those kind of cells. Okay, few more. Some environmental things are there. Tissue culture, we did these two, ne? Tissue culture and these two. Why do we say, so these environmental things, do I need to discuss? Yeah, I suggest we jump to some of the MCQs from molecular. Yeah, MCQs that we didn't do last week. Online people, which questions did we do last week? Did we do the final last time? We did do last time, I mean, from 40 to 50. Yeah. Did you do the animal? I think we did. Eh? We did this one. I can remember. This one I also we did. The genetic things we didn't do neither. Yeah. Online people, let me know. And also micro. I think from 31 onwards we didn't do neither. Yeah. After 45 we didn't do. Okay. What about 31 onwards? Did we do? Give me one. Yeah. Okay. Shall we discuss a few from 31 onwards? Yeah. 31 onwards. Certain plant of a particular species, they are purple, and the other plants of the same species, they are white. explain the inheritance of the flower color. To explain, okay. So there are purple flowers and there are white flowers. So what can you say about their inheritance? A monohybrid cross is sufficient, a dihybrid cross is sufficient, monohybrid and dihybrid cross is necessary, incomplete dominance is necessary, knowledge of gene linkage is necessary. What do you think? Yeah, don't overthink. They are given only one feature. There are purple color flowers and there are white color flowers. So, what is the only feature? Just one character is given, no flower color. So, do you need to think dihybrid crosses, dihybrid crosses? Do you need those? No. You are doing a dihybrid cross if you are following two characters at the same time. No. For example, flower color and the seed shape or the length of the plant. If you are following two characters at the same time, then of course you are going to need something dihybrid. So, by doing just a monohybrid cross, you draw a Punnett square, then you can identify, okay, for example, capital P, capital P, capital P, simple P, ne? Purple, okay? Then there's going to be capital P, simple P, simple P, simple P, ne? Yeah. If you have complete dominance, what are the things you are going to observe? If you have complete dominance, what are the things you are going to observe? Can this one, of course, purple, ne? Capital P, capital C, purple. No? What about this one? If you have complete dominance, that means capital T is completely controlling. Then math, purple. Eh? Yeah. What about this one? Is also purple. Eh? What about the this one? Is going to be white. Now tell me, if you have incomplete dominance, if you have incomplete dominance, what do you mean by incomplete dominance? What happens in incomplete dominance? Yeah. Mix characters. Yes. In incomplete dominance, there's going to be a mix of characters. So instead of this purple color, could be purple and white mixed, sort of a light purple, light purple. Yeah, purple and white mix in this capital P simple P situation. We don't know, right? So by doing just a monohybrid cross, you can say, okay, incomplete the gene linkage, whatever, just a monohybrid cross is enough. Don't overthink. Yes. Okay. We'll do one more. Yeah. Non-coding sequences and DNA segments. We'll do until 35 right? 34. Non-coding sequences and DNA segments without any identified function in the chromosomes of eukaryotes. So, who are the non-coding? This is a very nice question. And without any identified function. Non-coding sequences means they are found within the gene, but they don't code for protein, right? If you take a eukaryotic gene, it has pieces that are coding for protein, it has pieces that are not coding for protein. Non-coding sequences are called what? Introns, yes. Very good. Then DNA segments without identified function, they are usually found what? They are usually found, if this is gene 1, this is gene 2, they are usually found in between genes. Ne? So you call them intergenic DNA. Ne? So introns and intergenic DNA is the answer. Okay. Make okay. Select the correct statement regarding synthesis of polypeptides. Okay. Except for having U in mRNA instead of T in DNA, base sequence of the DNA template and its mRNA molecule is completely similar. What do you think? Is it similar, the DNA template and the mRNA? Is it exactly similar? No, you are making complement, right? So if you take the two DNA molecules, okay, if you take the two DNA molecules, two DNA strands, I mean, sorry, the two DNA strands, if you are making an mRNA of this one, if you are making an mRNA of this one, it is actually an exact copy of the other one, no? Yeah. So if you take the template, it is the mRNA that you are making is a complementary copy, no? So you can cut this end, right? The DNA templates and mRNA are complementary, no? Complementary. Yeah, this is true. In mRNA, you don't have thymine. That part is correct. Okay. An mRNA molecule of a prokaryote cannot use a polypeptide, can code a polypeptide in a eukaryote. What do you think? No, it can. It can, because why? The genetic code is universal. So you saying you cannot is false. Okay, sorry, it's false. Yeah, from a prokaryote or whatever, because the genetic code is universal, you can use their mRNA. Start codon of an mRNA is AUG. It codes for methionine. What do you think? Yeah, that is correct. No? I want you to remember protein synthesis starts in August with methionine. August will patangan. AUG, you can write here. AUG is the what do you call it? Start codon. AUG is start codon. Okay, AUG is start codon. There are 64 codons and 62 of them provide codes? No. There are 64, yes. Out of them, how many are special? There are 3 stop codons. Then one is a start codon coding for amino acids. So you have 61 coding for amino acids. There are 3 stop codons. So what are the 3 stop codons? UAA, UAG, UGA, UGA, UA, UEG, UGA. Sorry, UGA, UGA. Right? Just an easy way for you to remember, okay? The first triplet of bases in a tRNA molecule is AUG? No, right? A tRNA is coded in a different way, okay? So an mRNA will start with the AUG because then it will code for methionine first, okay? So false. Okay, any questions? So, okay, so this Friday, the procedure is going to be like this. We'll be starting the 2022 paper discussion. As usual, we'll be starting from plant form and evolution and animal form questions. And I will try to give 2 guest essays as well. And we can practice a bit of essay writing practice with those as well. Okay, so we have at least 6 more days of classes to do before your exam. I mean, before I stop this program that we have been doing. Okay, so we have covered a lot in the revision class, so that now you are stronger than ever to face the exam. So that assurance you need to have always, because in these final 3 weeks, that assurance is going to make you keep going. Okay, so try to make the best out of the exam that you are going to face this time. Okay, because it could be the last time that you need to do. Okay, the A-level exam is best done only once. Okay, right? So okay, then we'll stop for the day. Any questions?